key to act fast, and an aggressive intervention is needed to minimize the brain damage induced by hypoxia ischemia. The only treatment available nowadays is therapeutic hypothermia. During this treatment, newborns are cooled down below homeostatic temperature, typically to 33 to 34 Celsius, using a cooling cap or a blanket for 2 to 3 days, as you can see on the pictures on the right. The cooling decreases the metabolic rate and stops the spread of brain damage, but despite being treated with the therapeutic hypothermia, a considerable proportion of newborns still suffer from the consequences of hypoxic ischemic insults and develop some degree of disability. Another limitation of this therapy is that it has a limited time window of approximately 6 hours after the hypoxic insult. This means that neonates will not experience, or will not probably experience, an improvement from the treatment if the protocol is initiated after 6 hours. The prognosis of hypoxic ischemic encephalopathy will depend on the extent of the neonatal hypoxic injury and the possibility of initiating the treatment as soon as possible. And children with disability associated with this encephalopathy typically require medication and lifestyle support, and this highlights the high unmet need for treatments options for this condition. In this context, the study of epigenetics can provide us with new therapeutic tools to treat neonates with hypoxic ischemic encephalopathy. So first, what is epigenetics? And epigenetics is the study of how the environment can affect the way our genes work and express. DNA and the proteins DNA is wrapped around that are called histones, that are the blue balls in the image on the left, can get chemical tags that are epigenetic factors, here in yellow in the image. And these epigenetic marks can change the structure of the DNA and boost or interfere with the expression of different genes. As an analogy, you can think of switches that these epigenetic modifications can turn on and off to model the gene expression. This epigenetic regulation plays an essential role during development, and any disruption of the normal plan of epigenetic programming will have consequences. For example, an hypoxic insult will affect these processes. The main mechanisms of epigenetic regulation are DNA methylation, non-coding RNAs, and histone modifications. So considering this, the aim of this thesis was to evaluate the epigenetic mechanisms of hypoxia ischemia, brain damage, and the epigenetic mechanisms mediating the neuroprotective effects of therapeutic hypothermia. Because understanding these changes, we will be able to have a better understanding, to have new tools to treat patients with hypoxic ischemic encephalopathy. For example, we can use epigenetics for identifying new biomarkers or new therapeutic targets. So how we study this condition? In my case, I have made use of animal models. There are several animal models for studying hypoxic ischemic encephalopathy. I have used two of the most common rodent models. One is a neonatal hypoxia ischemia model, and one is the one below is the perinatal anoxia model. The one on the top mimics hypoxic ischemic condition, and that model mimics the hypoxic insult by submerging the term uterine horns of a pregnant rat in a saline bath, and the one below uses postnatal day 7 to 10 neonate pups and submit them to an anoxic chamber to induce the anoxic insult. Both models are good for mimicking the hypoxic ischemic insult and have some advantages and disadvantages. For example, the one below has the possibility of standardize more the stimulus and has fewer stress-related variables and lower mortality rate. But both models can induce the main hallmark metabolic processes that we can see on the clinical of humans with hypoxic ischemic encephalopathy as metabolic acidosis, hypoxia, and hypercapnia. So after the recovery of the hypoxic insult in both models, I have used treatment with normothermia or either normothermia or hypothermia to induce and modelize the therapeutic approach used in the clinic. And I have studied in neonates and adult animals the behavioral changes induced by either no treatment or the treatment with hypothermia, with a focus on studies on the cerebral cortex and hippocampus of these animals. Now I will present some results from these studies, and the first one, I'm going back to the epigenetic side. Epigenetic regulatory complexes are master regulators of gene transcription, and it has been postulated that epigenetic regulatory complexes may play a role in neonatal hypoxic ischemic injuries. In this context, the REST complex is a main regulator of neurogenesis and neuronal differentiation. And in chapter 3 of my thesis, making use of the perinatal hypoxic ischemic model, I evaluated the effect of hypoxemia and hypothermia on the REST complex and related gene expression. Results of this study on the right panel show that perinatal hypoxia, ischemia, induce an increase in protein expression of 3 components of the REST complex. REST, FOREST, and LSD1, and that peaked at 12 hours after the insult. And interestingly, the hypothermic treatment after hypoxia prevented this protein expression upregulation. And this concept highlights the therapeutic potential of targeting epigenetic machinery as the REST complex for the treatment of hypoxic ischemic encephalopathy. In the set of animals, I evaluated gene expression of these epigenetic components. And as you can see on the top graph, there were no changes identified, but that may be an bias by the selection of reference genes. So we made a publication emphasizing the selection of reference genes in the studies of gene expression to increase the accuracy and reproducibility of gene expression studies. In chapter 5, I evaluated the effects of therapeutic hypothermia in a rodent model of oxygen deprivation, and results show that therapeutic hypothermia reduced the anoxia-induced behavioral impairments, specifically using the fear conditioning task in adult animals. I showed that hypothermia reversed the impairments on risk assessment, behavior, and acquisition of spatial memory. So to explore why memory was impaired by hypoxia and restored by hypothermia, I studied the hippocampus, that is a brain structure related to memory, and results show that therapeutic hypothermia reduced anoxia-induced hippocampal cell death in the dental gyrus. As dental gyrus is a region of the hippocampus that is where neurogenesis takes place, and neurogenesis is regulated by a polycomb complex. This is another epigenetic complex, and the main regulator of polycomb is day2. I studied the protein expression of this protein in the dental gyrus, and in this case, we didn't find a correlation between the therapeutic benefit of hypothermia and this protein. So possibly it's not related to the neuroprotective effects of hypothermia. So to finalize, the main conclusions and impact and future directions of these studies are that RES complex proteins are modulated by perinatal hypoxia ischemia and therapeutic hypothermia and could be a valuable target to direct new therapies to. Therapeutic hypothermia prevents neurodegeneration in the dentate gyrus of rats submitted to neonatal anoxia. Hypothermia decreases EZH2 protein levels in the hippocampus of neonatal rats, and hypothermia may influence also physical development of rat neonates. So this highlights that therapeutic hypothermia may have side effects. As a future directions, biomarkers, it is a possibility to use epigenetic factors as biomarkers on one side to identify hypoxic ischemic encephalopathy at earlier stages, and on the other side to stratify patients to make the available therapies for hypoxic ischemic encephalopathy more effective. And on the other side, we can use these epigenetic complexes. A better characterization of these complexes will lead us to the development of new therapeutic tools and approaches to target HIV. With this, I thank you for your attention, and I will now give the floor back to the prorector and open it up for Q&A.
Thank you very much for this presentation. The opposition will be started by Professor Lesch. He was the member of and chair of the assessment committee, and he's from the Institute of Psychiatry of the Maastricht University and of the University of Würzburg. Professor Lesch.
Thank you, Pro-Rector, for giving me the word. Dear candidate, also thank you for this concise and clear presentation. Since I am the first opponent, I will keep my questions general, and I will also ask questions, primarily questions from the clinical perspective. Your work was on the neonatal hypoxic ischemic encephalopathy. You were referring to neurological deficits. What are the implications for the development of psychiatric disorders?
Thank you very much, Professor Lesch, for your question. It has been established in epidemiological studies that there is a correlation between perinatal insults and the development of psychiatric disorders. There is a correlation at the epidemiological level. The molecular aspects are still being elucidated and studied. In chapter 3, my intention was to identify that specific correlation studying the microRNA 137. That is a microRNA that is associated to schizophrenia, and schizophrenia is also associated to the hypoxic ischemic insults in the perinatal period. The correlation is there. That is a candidate gene, and that gene, microRNA 137, is regulated by the REST complex.
I would like to hear more about the clinical aspects. For instance, epidemiological evidence for impairment of cognition. As you may know, impairment of cognition is a risk factor for a wide range of psychiatric disorders, including neurodevelopmental as well as adult psychiatric disorders. What is the evidence for impairment in cognition?
Again, I think the main evidence comes from human studies at the epidemiological level. These perinatal insults have been associated to a range of neurodisabilities, and because the causes of hypoxic ischemic encephalopathy are diverse, the consequences are also ranging from anxiety to Alzheimer's disease. It depends on the impact and severity of the insult.
What about neurodevelopmental disorders, including autism spectrum disorders or attention deficit hyperactivity disorder?
This correlation has also been done at the epidemiological level, and epigenetic factors as, for example, methylation of certain candidate genes that are specific for these conditions can be induced by the perinatal insult at the perinatal period.
If I would be a parent of a child suffering from neonatal impairment, what would you give me as an information? What is the risk for neurodevelopmental disorders as mentioned earlier? And what could be done as early intervention to help the child to improve its development?
So you mean after the child has had the hypoxic ischemic insult, of course?
Yeah. What is the risk for autism or ADHD in the numbers? Yeah. As compared to other risks. You know, the incidence and prevalence of ADHD is about 10% in the population, which is very high. Autism is much lower. Would this increase the risk for 1, 2, or another 10%?
I don't have the specific number. I think that that will depend on the severity again and the cause of the insult. Hypoxic ischemic encephalopathy can be subclassified as moderate, mild, and depend on this classification, infants will be eligible, for example, for the treatment with therapeutic hypothermia or not.
Okay, let me move to the next question because there's time constraints. You were listing and talking about the different molecular mechanisms of epigenetic regulation, such as methylation, histone modification, microRNAs. I would like to hear more details about, for instance, about methylation of the genome. Where does it happen? Is it associated with genes or with other mechanisms? Could you allude to that a little bit in more detail?
So where methylation happens in DNA can be methylated or demethylated, and this will be affecting the expression of the genes.
Is it associated with methylation, demethylation of transcriptional regulatory regions, or with coding regions or non-coding regions? What is your opinion about the prevalence of these different locations in the genome?
So methylation happens at regulatory regions of genes. Yeah.
Okay. You were finding evidence for an increase of microRNA 137 in therapeutic approaches. Can you give me some more details how microRNAs are interfering with gene expression? There are several mechanisms. Could you just mention those different mechanisms and how microRNA 137 is actually acting?
The main pathway for an microRNA is it binds to the messenger RNA and it gets degraded. And in doing that, it impedes or stops the gene transcription.
Okay, but there are different locations again in the structural encoding of a gene in the genome. Is a microRNA binding to the transcriptional control region, or is it binding to the 3' untranslated region? Can you differentiate these mechanisms a little bit?
Yeah, so the main and most studied form is binding to messenger RNAs, and the other way is regulating non-regulatory regions.
Right. So you were referring that microRNA 137 is associated with schizophrenia, so psychosis. Coming back to the clinical aspects, what is the risk associated with this if there is, for instance, a decrease or an increase of expression of this microRNA? And can this microRNA also be epigenetically being regulated? There must be a regulatory region of this microRNA encoding. So is there also methylation of this?
Because of time,
Yeah.
dear candidate, I kindly ask you to answer briefly so we can move on afterwards to the next opponent.
Okay. Good.
Yeah, in that case, for that specific microRNA, the association with the schizophrenia is that these patients have been identified to have polymorphisms of this microRNA.
Right.
And this
okay.
polymorphism.
Thank you. This is okay.
Thank you.
I'm now giving back the word to the pro-rector. Thank you very much.
Thank you. The opposition will now be continued by Professor Michetti from the Department of Neuroscience of the Università del San Cuore in Rome, Italy. Professor Michetti. You are still on mute, Professor Michetti. Sorry, apologies, Professor Michetti. You are still on mute. Yeah, there you are.
Yes, but in my it is open. Do you hear me now? Okay, thank you. I want to thank you, the pro-rector, for giving me the word, and also the candidate for here really stimulating talk, clear and stimulating talk. And it is really stimulating. I have my first question deals with a class of proteins that maybe you have not studied. I don't know if you have studied. That is the cold shock proteins. You mentioned these proteins, I think, in the introductory part of your thesis, and they are really stimulating. I don't know if you studied them. It seems no, but I don't know. And in particular, I would like to receive some more information concerning, it's really stimulating, the hypothermia mimetics. May I ask you what you mean about that? And some more details about the behavior, if you studied, about cold shock proteins in this model.
Yeah, thank you, Professor Michetti. Yeah. I have not included any publication on my thesis regarding the hypothermic mimetics or cold inducible proteins, but I have experience on these proteins. In fact, I have collaborated with other students from my PhD director, Fabian, and the other publication was on this kind of proteins called induced proteins. Are proteins that are overexpressed under the exposure to cold. And those proteins, for example, SIRP and RBM3, RNA binding motif 3, and cold induced RNA binding protein, have been shown to have neuroprotective effects. In our publication, we have studied that, again, is not included in my thesis because it was other topic. We investigated these proteins for ocular trauma on the eye. And the idea was to create a mimetic of these proteins to be administered by as a drops, so it can protect traumatic injuries to the eye by inducing the expression of the genes under the control of these cold inducible proteins, but without the hypothermic or cold therapy. I don't know if that answers your question.
Indeed, it's a mimetics of hypothermia. That's very interesting indeed. Yes.
Yes, that is under investigation, and there are some products still under investigation. I think none has reached the market yet, but it's a really interesting approach for eliciting the neuroprotective effects of hypothermia in these conditions. One can elucidate that this would also be useful for other clinical conditions that have brain damage induced by, for example, hypoxia, as stroke, or any concussion on the brain.
Yes, it appears to be very stimulating indeed. Yes, thank you. My second question deals with, I think it's chapter 3. You speak about rest proteins. You investigated indeed rest proteins. It does not seem to me that you did any, let's say, immunohistochemical or morphological study. So my question and my curiosity deals with the cell location of these proteins. Do you have any information about that?
The cell location of the rest complex.
Rest proteins.
So rest at the cellular level is mainly concentrated in neurons and neuronal tissue, and at the cellular level, it's in the nucleus because it regulates the expression of the genes.
And it has not shown any, for example, cytoplasmic nuclear translocation of these proteins during treatment. I don't know, it's just a curiosity about that, but might be interesting.
I'm not sure. I have not studied that part of the regulation of these proteins. Yeah.
Thank you. So they are specifically located in neurons, not in glia, not in astrocytes, for example, or other cell types. They are specifically neuronal proteins, and in particular, nuclear neuronal proteins. Okay. Thank you. I may come back to the prorector if he likes.
Co-rector, you have to unmute. Apologies. Thank you, Professor Michetti. The opposition will now be continued by Professor Homburg. She is a professor in translation on neuroscience of the Radboud University Medical Center from this country. Professor Homburg.
Yes, thank you. I think you can hear me. So, dear candidate, to start off, I would like to congratulate you with your thesis. Also, my compliments for your promotion team. Of course, they supported you a lot. I was really pleased to read your work. It was for me also a new topic, so I learned a lot. And I think it is also a very relevant topic because, it really has clinical implications. And we know, of course, that a healthy start of life has consequences for the entire life. So very important topic. But of course, I'm not here only to just give all kinds of compliments, but also to ask some critical questions. So I read your thesis and, I understood you were using rodents to induce the hypoxic ischemic, and so I have to practice on the words, encephalopathy. And you did that with female rats that were just delivering their offspring. And I had some questions about it because it is known that rodents are born immature compared to humans. So around postnatal day 10 or 11 in rats corresponds to birth in humans. So I wanted to question whether your chosen model is really a good model for the human situation because of that.
Thank you, Professor Humber. Yes, in fact, I have used two models, and that highlights this specific topic of the delayed development of the brain of the rodents versus the human. In the perinatal hypoxic ischemic model, that is when the pups are still in the uterus, the hypoxic ischemic insult is more similar to the human one because we are interrupting the blood flow to the pups. But at the same time, it has the disadvantage that it mimics a very low gestational age of the human neonate. Mm-hmm. On the other side, the other model has the benefits of mimicking the anoxic insult at the gestational age of a perinatal human. That will correspond to postnatal day 7 to 10 in the rat. And that allows us to study, for example, neurogenesis in the hippocampus, and it will be more comparable to the human. But in this case, we don't have the ischemic component of the insult. Okay. So both are models and both are complementary, I would say.
Yeah, okay, clear, thank you. So in chapter 3, so you had you mentioned schizophrenia as one of the consequences. And I also read that, so you applied the hypoxia as a treatment, and I read that the pups were decapitated 2, 6, 12, and 24 hours after the hypoxia treatment to assess epigenetic markers. But I did not understand why these time points, because schizophrenia appears during adolescence, right? So how can this information be informative for schizophrenia?
So the idea of studying different time points was to know or describe better how these proteins react, and because the hypoxic ischemic insult is abrupt insult, the reaction, the physiological reaction will most likely have a short time span. So we were not sure on at which time point we will capture or read the change in these proteins. That was why we chose 2, 6, 12, and 24 hours to study the gene and protein expression of these proteins. Okay. And that also helps us to understand if these proteins have indeed a therapeutic value, because if those are induced, I don't know, 1 week after the insult, most likely the damage is already caused, and those don't have a therapeutic value.
Okay, but do you agree that it might also be interesting to have longer time points into adolescents and even perhaps adulthood?
Yeah,
Because you
of
also
course.
would like to know what the long-lasting effects are, right? Yeah.
I agree. Complementary studies, including behavioral changes and correlating behavior with gene expression and epigenetic changes, are still useful and needed.
Yeah. Okay, thank you. Also, perhaps a simple question, but still relevant. So I was wondering, did you also discriminate between male and female pups? When
Yes.
doing, yeah?
Yeah.
Okay,
all
can
the
you do
studies-
that really with newborns, or can you really already dissociate the males from the females?
Yes, we can discriminate newborns by sex, and we use only male rats.
Okay, but might also be interesting to include the females, right? Perhaps in the future, because I guess there
Yes.
is there a sex dependence or for the ischemia, or does it happen to both boys and girls?
Usually, the behavioral studies are conducted in male rodents because of estrous cycle of the females. Rodents can affect the behavioral response and in the end the results interpretation. So it has been established that the male studies is the way to go. But yeah, I do agree that including female animals will be the next step to improve.
Yeah, because
our
I also
understanding of
think that pups don't have an sex yet, right? So
No, but some differences, physiological differences can also. No, but the idea was to maintain those animals until adulthood, to
okay, yeah.
some of those animals to until adulthood to use on behavioral studies.
Yeah. Yeah, but I think it is a little bit, if I may say, a little bit old-fashioned thought nowadays that the cycle of the
It is.
females has interference, because there is no evidence for it, and
It
diseases occur also to females. So we are lacking behind with information on females. So
I agree. Yeah, I totally agree.
So another question is, so in chapter 3, you focus on epigenetics in the cortex, so you punch out the cortex, but in chapter 5 on the hippocampus. I was just curious, why did you choose for different brain areas across the different chapters?
The idea was to study different processes. In the one I included with the study of the hippocampus, that was a collaboration with an institute in Brazil, in Sao Paulo, and they were studying the hippocampus and memory and the impact of the anoxic model and hypothermia in memory. So that was why I switched to the hippocampus in that one. In the beginning, my original project was, the idea was to study cortical layering of neurons. So how neurons establish different layers on the cortical cortex and how that is influenced by microRNAs, for example, 137, that is associated to schizophrenia.
Okay, thank you. If I still have time, then I continue with the question. So in your experimental design, where pups are being exposed to hypoxia plus hypothermia, they are placed with foster mothers, and that is logic because the own mothers are being killed. But yeah, of course, that is not a situation that happens in humans. So I was just wondering whether the change in mother itself can already cause epigenetic changes. Perhaps the animals notice somehow unconsciously, like, this is not my biological mother. So what is your view on this?
You mean placing the animals from the hypoxic ischemic model with a different surrogate mother, right?
Yeah.
Yeah, I guess that is also a variable of the model that is a negative side of that model. In fact, that model has that, and then the surgical procedure of having the C-section on the first rat, and so from all the processes that those animals receive, having the surrogate mother, I would say, is the less severe, but for sure it also has some consequences.
Okay, thank you. Yeah, I'm not sure whether I have time left over. I can also give the word back to the rector. Okay, my time is over. Well, thank you.
Thank you.
Thank you very much.
I think you are on mute.
Apologies, he keeps flipping on mute. The opposition will now be continued by Dr. De Nijs from the Department of Psychiatry and Neuropsychology from the Maastricht University. Dr. De Nijs.
Thank you, Pro-Rector. So, dear candidate, I would like to congratulate you for your nice thesis that I enjoyed to read and your nice presentation. Also, congratulations to your promotion team. So I will try to keep it short because of the time. And I had some question about your chapter 3, where you analyze the expression of protein and messenger RNA of different subtype of rest complex at several time points, but I will focus on 24 hours. And in figure 1b, you observe a 5-time increase in rest at the protein level. However, on the contrary, at the same time point, you don't see any changes in the mRNA level, so in figure 2b. So how could you explain these differences in protein and messenger RNA expression at the same time point?
Thank you, Dr. De Nijs. Yeah, that was an issue on the interpretation of those results in chapter 3, and that's why we included an interpretation related to the selection of reference genes that can affect the understanding of the results obtained of qPCR gene expression studies. This is a critical part of these studies because it affects, of course, all the results and affects the reproducibility and validity of the results in the end. That will be an way to explain the difference and discrepancy of mRNA versus protein, and the other one can be degradation at the protein level of the rest protein. Degradation can be stopped somehow by the therapeutic and the epoxic insult. So that can also be the other option.
Yeah, so thanks to develop this, because this brings me to my second question. Indeed, you nicely analyze in your chapter 4 that different housekeeping gene change in expression between the different group and different condition, and you were on to test several housekeeping genes and find the good ones suitable for your study. But in this chapter 4, you indeed found out that, for example, the U6, I will simplify it to U6, is increased with hypothermia, and I have found all the studies in literature showing also in heart muscles that U6 is increased with hypothermia, but you still use it as normalizing gene for your microRNA in chapter 6. So how can you trust then the data you show in chapter 3 by showing an increase of microRNA 137 by hypothermia itself? Do you trust this, or what would you do then to improve this data?
Yeah, that was an issue, and I think that's the main issue for chapter 3 still not being published, that the expression of MIR 137 will be depending on RNA U6. One way will be to include more reference genes and then choose the more stable one to assess the gene expression. I think that will be the best option for that.
Yeah, thank you. So a last question, I would like to jump follow-up of Professor Lesch's question. So you indeed explain in the chapter 3 introduction that schizophrenia is associated. So hypoxia in infants is a risk factor for developing schizophrenia, but also a risk factor for schizophrenia is a change in expression SNPs in this microRNA 137. And as you studied as microRNA 137, I would like you to develop a little bit how you do see the mechanism at the molecular level, more how expression of this microRNA 137 could later on then lead to the development of schizophrenia. So how do you see and which mechanism could be impacted and based on what you study here as maybe just a reflection?
Yeah, the idea for that study was the correlation between that microRNA and schizophrenia is a polymorphism, or that reduce the level of activity of that microRNA because it has less affinity for the target genes that it should silence. And the idea was to check if the epoxic insult was enough to reduce the activity of this microRNA in a critical period, that is the postnatal development, when cortical migration of neurons occurs. And this specific process of cortical neuronal migration is regulated by this microRNA. Cortical migration also, it has been demonstrated that is impaired and is disrupted in schizophrenic patients. So that is the relation.
Okay, thank you very much. So I'm happy with the question, and I give the words back to the pro-rector.
Thank you.
Thank you. The opposition will now be continued by Dr. Gazzolo he is a professor in neonatology and pediatrics in Chieti, Italy, and this university. Professor Gazzolo.
Yeah, Director, thank you for invitation. And I carefully read the thesis of Dr. Bustelo, and it's very interesting, and the promoter has to be congratulated for this thesis. Anyway, my question is mainly based on the future of his perspectives, of his studies. And I wonder whether in neonatology, the time is the keyword for this kind of procedure. Hypothermia is a life-threatening procedure for asphytic babies. So from a laboratory point of view, I want to have these two questions. The first one is the time point for laboratory performance for an epigenetic test. How much is the timing that the laboratory spent to provide results output? And in the last chapter of relevance and the future perspective of laboratory, of also biochemical assessment in this kind of pathology, is stated that the standard of care procedures such as a clinical score, Apgar score, EEG, and clinical evaluation cannot be available for deciding which are the babies that can be treated by hypothermia. I would like to know the future perspective of epigenetics tests in terms of clinical decay and the usefulness for future babies management. Can you hear me?
Dr. Yeah, I can hear you. Thank you, Dr. Gazzolo. So the question is, what are the clinical implications of the epigenetic components for HIE, right? So as I see it, the first and most probable use will be to establish hypoxic ischemic encephalopathy risk in pregnant females. And in doing that, we can secure that these neonates receive the therapeutic hypothermia treatment on time, and to improve the possibilities of them to have a better outcome. The other application of epigenetics will be creating, for example, panels that can tell us better than the standard of care, than Apgar score and the current clinical assessment, how bad is the hypoxic insult that the neonate has. So that will be epigenetics will help us to stratify this population between mild and moderate candidates to receive therapeutic hypothermia or other adjuvant therapies.
Yes, but sorry if I stop you. But the first 6 hours for us are crucial, since we have to decide which of the babies are suitable for hypothermia. So how long lasts an epigenetic test in order to obtain the results, in order to decide this from an epigenetic point of view for this parameter is suitable for hypothermia or not? This is a question.
So there's still no commercial or any type of available epigenetic or metabolic biomarker used in the clinical side. My guess it will be that any biomarker should be easy to collect, as for example saliva, plasma, and easy to analyze and quickly to obtain results before 2 hours of birth, right? To secure that the therapeutic hypothermia protocol can be initiated as soon as possible. So those are the two main qualities that any biomarker should have.
Okay, thank you. And based on your studies, we know that prolonged hypothermia can be dangerous, is not neuroprotective, especially from cardiac surgery experiments also and in clinical practice. So also the lasting of hypothermia is an issue that has to be taken into account, especially from the degree of temperature, because this in clinical practice we have mild hypothermia up to 33 degrees of cooling. So based on your experimental and biochemical study, what do you think about the hypothermia performance? You agree with what are now the standard protocols, or do you think that we can improve also from the temperature modalities of performance?
I think there is still space to improve the protocol, specifically not only on the temperature side, but also in the inclusion criteria of neonates. For example, some mild neonates with hypoxic ischemic encephalopathy with a mild degree, but that are bigger, may be eligible. So I think the protocol and making use of new biomarkers can be more targeted and creating a better outcome using this treatment.
Thank you, Director. For me, is okay. And if you go further,
thank you very much. Thank you very much, Professor Gazzolo. The opposition will now be continued by Dr. Barkhausen from the Department of Computational Biology, Germany.
Dear candidate, thank you for your thesis. I wanted to know whether the rest proteins would be suitable as biomarkers for hypoxic ischemic encephalopathy.
Thank you, Dr. Barkaisen. The idea of this experiment was to study the rest complex expression in the brain. I have not studied in any biological fluid as a biomarker. That will be really interesting to study. I have no knowledge on the study of this specific protein in any biological fluid that can indicate that it can be used as a biomarker. I see this protein more as a therapeutic target because it's one neurological specific and other it's a gene regulator. So maybe as a biomarker, I will use metabolic indicators or molecules, and this can be more as a therapeutic target.
Is there any things that you studied in your thesis that would be a suitable biomarker?
Sorry, can you repeat?
Any of the molecules that you studied in your thesis a suitable biomarker?
As I see, microRNA 137 microRNAs can be probably used as biomarkers. 137 is also neurospecific, but other microRNAs can be candidates better than epigenetic complexes to be used as biomarkers.
Do you know it's detected in the blood?
Not 137, but for example, microRNA 210 has been proposed as a clinical biomarker of hypoxic ischemic encephalopathy and brain damage, and it is also correlated to the clinical outcomes of the hypoxic insult. So not that one, but there are also other microRNAs that.
No, thank you for your answers, candidate.
Martin Bustelo, the time
Thank you.
appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations until our return. Thank you very much. Martin Bustelo, the degree committee here present online has assessed the quality of your thesis and your defense. In view of this positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Steinbusch is authorized to confer you this academic distinction in accordance with the Dutch university custom. I invite your supervisor to take now the floor.
Thank you very much, Mr. Prorector. Dear Mr. Bustelo, first of all, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful, to be honest, transparent, independent, and responsible? You have to answer now.
Yes.
You have to say, yes, I promise.
Yes, I promise.
Thank you. Okay, thank you very much. So, because it's all recorded, yes, of course. So by the authority vested in us by law, and in conformity with the decision of the committee here present online, I hereby confer upon you, Mr. Martin Bustelo, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon, not now, soon, receive the degree certificate signed by the rector, signed by the secretary, and signed by the supervisor affixed with the official seal of our university, and it will be shown now to you by the beadle. Yes. So please be in mind that the certificate will be signed to you, and we will send it to you by DHL or whatever, all the way to Argentina. And by that, I would like to give the word to Dr. Gavilanes, who now will address you in a personal way. Professor Gavilanes, I give you the word.
Thank you, Professor Steinbusch. Dear Dr. Bustelo, dear Martin, as we call you here in the Netherlands, but estimado Martin, you are Martin for me. Well, I would like to congratulate you for the way you defended your dissertation and for the quality of your thesis. It was really an honor to have been part of this international multidisciplinary supervision team, which allow us to know you for the last 6 years, since 2016, actually, yes. A quite unique period of time, including the pandemic. Well, your projects and scientific output in general show your primary commitment to neurology and translational research. Second, you have worked and developed several role models of neonatal encephalopathy, working in different labs, in different countries, Argentina, Brazil, and the Netherlands. And you are a dedicated researcher. You worked on your thesis without neglecting your actual work activities, which are completely different, as I recently know. And in addition, the work you defended facilitates, and this is very important, facilitates future collaborations within institutions in Argentina and our university. And well, finally, I would like to extend my sincere congratulations to your family, and especially to your nonna, to abuela, which I don't know, but is the person to whom you dedicate your thesis. Martin, I expect that you will continue to develop a successful career, and I wish you all the best.
Dear Dr. Bustelo, on behalf of the Maastricht University, I also congratulate you with the degree you just acquired. And I would like to note for your family and friends that the degree of doctor is, according to Dutch law, the highest degree an academic person can obtain. So we also have professors or assistant professors, but formally by law, the degree of doctor is in this country the highest you can achieve. On the other hand, we all hope you just heard what Dr. Gavilane said, that it will not be this degree only, but that you will continue research, and we might even continue research with you in collaboration with you, I mean. So I also would like to congratulate your family with this unique degree. And hereby I would like to end this ceremony.